pleasure and privilege to welcome this standing room only crowd to the Airbnb mug renewal program. So what you do is you interview for a job, you have a kid or two, you break the mug, you bide your time, come up for tenure, you get a new mug. You have another kid, you break the mug. It was always full. After that, you're on your own, man. All right. This is a special moment in our department's history, in Jesse's career and life. He's worked really hard. I'm not going to waste his time introducing him. You all know where he comes from and what he's done and what he means to the department. This is your special day, Jesse, in front of your family, your community, your colleagues, your peers. This is your chance to show us what you've been doing, and I'm really looking forward to it. Jesse Goldberg. I thought this would be a good, is it a mug exchange? So, this is what a model system looks like. Yes, but in the ear of that finch are the microbes that run the world. Chemistry. That's right. Okay, thanks, Rob. Thanks everybody for coming here. Your support means a lot to me. And what am I? Good guess, Sandy, but that's not right.
That's not Sandy.
Because okay, there we go. Excellent. So my lab. Didn't test the set. Oh, good. We got sound. Okay. I'm interested in neural circuits for trial and error learning. And why is trial and error learning so important? It's because when we're babies, we really can't do anything. So basically, everything that you do, your whole behavioral repertoire, as a human especially, is learned. And I have a brief story to tell here. Many of you have heard it before if you take the intro neuroscience class, which is that this is Sandy, who's in the audience here, when he was about 7 months old. And whenever we wanted to neglect him, we would put him in the neglector saucer. You could put him there. He's safe, you can take a shower, you can wash your hands. And this particular neglected chair had a little handle here, shaped like a frog. And if you grab the frog, a little jingle would play. We put him in here, and every once in a while you'd hear the jingle, and he'd kind of be looking off in space, and his arms would be wiggling around. Then one day, Elizabeth remembers, we heard the jingle, and then we heard the jingle again, and we heard it again right away. And I grabbed my iPhone, and I'm like, I think he's got a goal-directed behavior. So I took this video, and you can see here, so it's not easy to reach for a handle and grab it, as you can see. He's got it. Now to do this, you have to have a target in visual space. You have to bring your hand to that target. In order to bring your hand to that target, you have to know where your hand currently is. Then you got to get it to this handle, and you have to contract your fingers at just the right space, get the handle, and then pull back. It's actually a pretty complex sequence. It's really hard to do. You can see he's struggling with it here. Can't quite get it. I think he's going to miss. Did he already miss? Oh, he got it again. So within hours, as you can imagine, we had to take the batteries out of this thing. And within a few years, he's running around, talking, climbing trees, playing sports. And I would argue it all starts with things like this. So the question is, how do we learn through practice? How does our performance in anything get better with practice? And the way that people tend, especially behavioral neuroscientists, often address this question in the lab is by leveraging the fact that animals will learn to do arbitrary things for food or juice, especially when they're hungry or thirsty. So this cartoon really is a lot of what neuroscience labs do when they study motor learning. And here's how a typical experiment works. Let's say you take a monkey and you're trying to understand how it learns to move right, left, up, or down. So you could have a monkey looking at a computer screen, And it looks, and the number 2 pops up on the screen. And he's got to figure out how to move a joystick or his eyes up, down, left, or right in order to get a juice reward. It's a thirsty monkey. So he guesses right, nothing happens. He guesses up, nothing happens. He's moving around, nothing's going on. And all of a sudden he guesses down and he gets water. He's super thirsty. Wonderful. And with practice, this animal can learn every time it sees the number 2, it just moves down and gets its water reward. Okay? And this is just basically classic stimulus response learning. So you have the stimulus, which is anything you're looking at the world. In this case, it's the number 2. The response, pull the joystick down, and then you get your water reward. Now, importantly, back in the 50s, when scientists such as Olds and Milner in Montreal were trying to figure out if there are parts of the brain that carry reinforcement signals, they basically put electrodes all over the brain, and they asked, will animals press buttons just to get some part of their brain stimulated? And it turns out that animals will press levers just to get a part of their midbrain stimulated that it was later determined contains dopamine neurons. So in a sense, animals are going around learning to do things to get dopamine stimulated. So dopamine can stand in as a reinforcer in a variety of behavioral paradigms, such as lever pressing, decision making, even place preference. And recordings from dopamine neurons in monkeys several years later in the 90s by Wolfram Schultz and colleagues showed that dopamine neurons exhibit a signal that's just perfectly suited to drive reinforcement learning. So this is the activity of a single dopamine neuron recorded in the midbrain of a thirsty monkey. And this monkey has learned that exactly 1 second after a conditioned stimulus or a tone he hears, 1 second later, he's going to get a drop of juice. And he's in this task, and he's trained in this task, and he's sitting there, and the tone plays at time step 0. And here what you see is the discharge rate of a single dopamine neuron, and there's a peak of activity right after the tone. The animal's like, yes, I'm about to get juice. This is called a positive prediction error signal because he didn't know the tone was going to come. The tone came out of the blue, and it told him he's going to get juice, and he's thirsty. So his state, the state of his being is better than he was expecting at that time step. So dopamine neurons are activated by events in the world that exceed expectations. So you can think of them as surprisingly good events. Okay? Now, in rare cases, what the experimentalists did is they withheld the juice. It's 1 second later. So the monkey's psyched, he thinks he's going to get juice, time passes, and at 1 second he's like, okay, I'm going to get juice. And most of the time he does. But in this particular trial, they don't. They don't give him the juice. And what happens to dopamine activity is you see it dips. Okay, so it decreases its activity. And you see these decreases in worse than expected outcomes. He was expecting a reward, he didn't get any. And this is called a negative prediction error signal. So you can think about this as a disappointingly bad outcome, which is associated with suppressions of dopamine activity. Now, the reason why these signals are so important is because this is thought to be the reinforcement signal that tells your behavior repertoire what should remain and what should be extinguished, right? So if there's a burst of dopamine, I need this laser pointer out, this is basically saying, it's projecting the parts of the motor system saying, hey, whatever you just did, do it again, right? And so all drugs of addiction, or at least most drugs of addiction, are thought to work primarily by activating dopamine receptors, or in some way activating the dopamine system, okay? Now, on the flip side, this suppression of dopamine firing is when something disappointingly bad happens, and in a sense, that signal is telling your motor system, hey, whatever you just did, don't do that again, right? And so if you lose dopamine neurons in a disease like Parkinson's, you wind up frozen. You don't move. It's like the signals that repeatedly told your motor system, don't do that again, don't do that again, don't do that again, and you become akinetic. So it goes without saying there's a lot of interest in these signals because in some ways, for those of you who think about evolution in the department, species evolve through time by procreation. Within an individual, behaviors evolve through time on the basis of having been selected, right? And behaviors extinguish through time on the basis of having been eliminated. And the dopamine system plays a huge role in which behaviors survive and which behaviors die within the evolution of an individual, okay? All right. So we also have a very good understanding of how this signal implements reinforcement by decades of work in the basal ganglia. Okay, so a little bit of background. Here's a little miniature circuit that's familiar to many students in the class, but to some people who don't know much neuroscience, maybe not. This is a basal ganglia circuit. I'm making it really simple. You can just think about it as part of your motor system that has different output channels. So here's a little pathway that can move your joystick up, move you up. Here's a pathway that'll move you down, and they're separate in space in the brain, okay? And Basically, what you have to learn in order to become an expert in this task is to hook up the representation of number 2, somewhere in your visual system, to the appropriate action, to move down. Okay? And here's basically how dopamine reinforces behavior. So you've got this system architecture. You've got 2 motor channels that move up or down. You've got representations of the world, such as what you're seeing, up in cortex, and they project down into both of the channels. And the dopamine signal comes in right at this synapse, or connection between these channels, from the cortex to the striatum, where these channels. So let's say you're in this task, number 2 appears. You guess up. I'm sorry, you guess down. Okay? Number 2 appears, you guess down. What happens when you guess down? You get juice. A better than expected outcome occurs, so dopamine neurons get activated. Now there's a special learning rule in the basal ganglia where if 3 things happen at the same time, then you change the connection strength between this neuron that represents what's going on in the world, like looking at the number 2, and this neuron that drives your behavior down. So this dopamine modulated corticostriatal plasticity rule, if these 3 things are active, you get long-term potentiation or synapse strengthening here. So that there's a physical change in your brain where the connection between number 2 and moving down is strengthened. Next time you see the number 2, you're not guessing down. But by virtue of this increased connection strength, you're actually the representation of the 2 is driving you down, and you get rewarded. So this is how dopamine modulated corticostriatal plasticity results in something we think of as like reward-weighted stimulus response associations. Okay, so basically you're maximizing your reward in this kind of paradigm. So this is like an ancient architecture. Insects have a similar architecture in the mushroom body. They have odor representations that connect to different motor output channels, and dopamine can weight these connections to make insects get drawn to or get repelled by different odors. So it's a really ancient architecture for stimulus response learning. Now, there's something deeply unsatisfying about this whole setup I've given you, which is that most of the things we do in life are not in pursuit of rewards, right? I'll give you an example. So here's Evan when he was just learning how to talk. And speech acquisition is known to depend on an intact basal ganglia. So kids with basal ganglia pathology in some way have impaired speech acquisition. And let's just take a look at what a normal kid, when he's babbling, is doing. As far as he's concerned, he's alone. He doesn't even know I'm secretly videotaping him. Okay, so there's a lot of things going on here. The most important thing is learning to talk. Kids do largely when they're alone. Okay, they babble when they're alone. There's no one giving him juice reward when he makes the right sound. There's no reward, so you can take that out of the equation. There's also no external stimulus that's dictating precisely what he should do at a given moment in time. There's no thing in the world that's saying this is how you should move your tongue right now to get reward. So it's very difficult to imagine how the dopamine basal ganglia pathway I just talked about for stimulus response learning that even a fly could do something like this. So let's just play a little game and just try to stretch our minds to see how it could work if we were to force that model of dopamine-dependent learning into a situation like this. So bear with me for a second. Let's pretend that he's trying to say a really fast car. And this is not such an unrealistic thing that he'd be trying to say. Because at this age, I could say, Evan, point to the fast car. Point to the really fast car in the book. And he would point to the Lamborghini and not to the pickup truck. So his auditory system could parse really fast car. He could represent really fast car, and he didn't even know what it means, right? But if he wants to say really fast car, he can't convert that auditory representation into a vocal motor sequence that matches the auditory representation. So let's just play this game and say, okay, he wants to say a really fast car. What he produces on one given moment in time when he speaks this one sentence is a really fast car. Okay. So it's pretty obvious that he makes a mistake. So he wanted to make a k sound here, but he made a f sound. There's a mismatch, so there's an error, right? Now importantly, there's nothing intrinsically bad about the f sound. It's not like the disappointment of not getting juice you expect. It just didn't match his expectation, or his desire, his target. Here, the f sound is just fine. Okay? So you would have to be evaluating your behavior to aligned sensory targets in time. A second problem is that we don't really know when he says a really fast bar what is good or bad to him, or what is good performance, because it might be the case, as was the case, that when he wanted to make the L sound, which is a hard sound to make, he made the W sound. He said really fast car, right? So it might be the case that in his past, he had never really nailed that L sound. So he said really fast car. But let's just say on this particular trial, he nailed the L sound. Some part of his brain guessed to move his tongue up just to the roof of his mouth to make the L sound. And a lucky guess it was, because it matched his target. Well, in that case, this L would be a positive prediction error. It was better than he was predicting, right? So, if you were to really buy into this, which there are a lot of reasons why you wouldn't, you would have to imagine that a dopamine signal that would be evaluating this type of behavior would do this. It would exhibit a peak in activity, or a burst, when performance was better than predicted, and it would exhibit a pause in activity when the performance was worse than predicted, because he usually would be able to nail the K sound, right? So it goes beyond speech. I mean, really what this would require would be that your dopamine system is not just evaluating your reward outcomes when you're hungry or thirsty, but that every millisecond of your life is being evaluated according to temporally aligned goals, right? So it wasn't known if this was the case. And the big question is, can we take what we know about dopamine-dependent reinforcement learning and see if what concepts can generalize to just all behaviors such as speech that require learning. Okay, so today I'm going to talk primarily about this question of performance evaluation with birds. And the reason why songbirds are such a good model system to address this question is because, in contrast to monkeys or rats or mice, you motivate these animals to do things in the lab for primary reinforcers, such as food, juice, or in some cases, painful stimuli such as shots. Birds just naturally learn to sing. You just put them in a box, they learn to sing. So they have a target they want to reach vocally. Okay, and so we can ask, do dopaminergic reinforcement signals apply to something like vocal learning? And then we're going to take a deep dive into the origins of these dopamine signals. And then hopefully at the end we'll have time to talk about what half of my lab is doing, which involves mice. And I'll do that briefly. Different questions of mice. Okay, so let's get into it. The song of an adult zebrafinch is a precise sequence of notes that we call syllables, A, B, C. And this x-axis is time, the y-axis is frequency in the spectrogram, and this is what an adult bird sounds like. So it might just sound like a scratch and Nintendo game to you, but to female zebrafinches, it's the bomb, right? And this is a mating behavior, okay? And these animals are showing off how good they are at learning. And what they're actually doing is quite complicated. So the sound is produced by bilaterally symmetric syringes, which are these vocal organs. Each of these syringes has 7 muscles. These are the fastest muscles in vertebrates, as far as I know. Maybe bats and songbirds have equally fast muscles. They can contract and relax on about a 5 millisecond interval. Let's give it 10. Let's say 10 milliseconds. About 5 to 10 milliseconds. Okay? And what that means is the song, in essence, is about a second long. You can imagine it being played on a 14-key piano that can be struck 100 times in a second. So imagine that's the song. It's 14 keys on a piano, and you got to strike those keys in just the right connection, just the right combination, 100 times in a second. And that's basically what the song is. Okay? And they produce it very stereotyped when they're singing to a female. Cha-chin-chin, 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 and the females evaluate the song on the basis of how stereotyped it is. Now that's an adult bird. But juvenile birds, they hear the adult song when they're like 30 days old, somewhere around there, and then you can remove these birds from the adults, put them in a box without tutors, and they'll just start babbling. And basically it's just like they're banging on the piano randomly. And when I say randomly, it's genuinely random. The syllable durations are exponentially distributed in these babbling birds, which is, as some of you will know in the audience, that's as random as a process can get. And when they start singing, they just sing this. So it's just basically scratchy, random sounds. and then give that bird about a month and a half, alone, in a box, and you hear that stereotype rhythm. He's matching the syllables in sequence and in phonological structure. Okay, so if you deafen a bird around this age, they can't learn to sing. So they have to listen to themselves. So the big idea, it's been around for a very long time, is that what practicing birds are doing is they're listening to their own song, they're comparing it to a target, and on the basis of that comparison, they're generating error signals that drive learning. Okay? And another reason why the birds are such a good model system to test these ideas of dopaminergic function is that it was discovered about 40 years ago that birds have a specialized vocal system in their brain. So this is just a slice of the bird brain, and you can see these structures here where there's increased cell density. They just jump out at you. These are nuclei that are interconnected into a circuit called the song system that is entirely dedicated to singing behavior. Not grooming, not eating, not flight, not landing. These are specialized vocal circuits, much like humans have. Okay? In fact, specialized isolated vocal circuits evolved independently in parrots, hummingbirds, humans. Okay? And about 15 years ago, about 15 years ago, it was discovered that this song system is part of just a general architecture of vertebrate brain where it has cortical, basal ganglia, thalamocortical loops, and importantly, there's dopamine projection from the midbrain into the basal ganglia. That people thought was important for vocal behavior. Okay? So, in order to test if that projection can carry something like a reinforcement signal that drives learning, Vikram Gadekar, Pablo Puzari, and Radon Chen, the grad student lab, working with Alex and Eliza undergrads, wanted to record dopamine neurons as we could trick birds into thinking they made errors or did a good job. Okay? And the first thing they had to do is record dopamine neurons. They implanted electrode arrays into the midbrain and stimulated electrodes into the part of the basal ganglia where these neurons project, and recorded from neurons in the VTA that projected this area X nucleus, which is the vocal motor striatum, part of the basal ganglia. And it was known from past work that this is a dopaminergic projection. So that's how we knew we were recording the dopamine neurons. And then we needed to trick the birds into thinking they made mistakes. So what we did was, the bird would sing a song ABCD, ABCD, and we would take a little snippet of C and analyze the song in real time, and we'd roll the dice, and half the time, we would play that snippet of C on top of B. So the bird wants to sing like chicken and here chicken, chicken. And it was previously shown by many labs in the song field, including my postdoctoral lab, that if you deliver such distortions contingent on how the bird sings a syllable, they will learn to avoid the distortion. So the distorted renditions, whatever is something is saying, hey, whatever you just did here, don't do that again. Whatever you just did here, here, do that again, right? So it was known that the distortions can act as error signals, can drive plasticity in the brain. Okay, so here's one of the first neurons that Vikram recorded. So on the x-axis here is time, and this is just voltage coming out of electrodes in the VTA in the brain of dopamine neurons, and these spikes represent dopamine activity. And what you see here is the distortion, and you'll see there's this pause in activity here. And here's 24 renditions of this song, and what you see is after the distortion shown here in yellow, you see this silencing of dopamine activity that lasts about 100 milliseconds. And here you see in the firing rate of this neuron, on average, there's this dip. This looks exactly like the dip that you'd see in a dopamine neuron of a thirsty monkey at the moment it doesn't get the juice it was expected to get. And on those trials, so we consider this as a worse than expected signal. Interestingly, on those trials where we didn't distort, so the animal heard the syllable just as he produced it, and you see here there's this nice harmonic stack, there was a burst of dopamine firing, which you can see here in this rate histogram as a peak in the dopamine activity. And this is exactly the kind of signal that a dopamine neuron would generate in a thirsty monkey at the moment it unexpectedly gets juice. Okay? And here's just another way of plotting this data, where 0 is the time of the target syllable. This is undistorted rendition, distorted rendition. There's the burst here in blue, and the pause, and there's another neuron with a big positive prediction error signal. So when we went into these experiments, we thought we might see this signal. We thought we might not. We didn't know if dopamine was actually doing anything related to evaluation. And it's easy to imagine why there's a pause here, because an animal wants to sing chickawa ant, and here's chickawa ch. There's obviously a mismatch, right? So it doesn't match the tutor song. So it's easy to imagine that this is a bad outcome. But we were really intrigued by these bursts because we're not doing anything to the song. The bird's just singing, right? So we hypothesized that what's going on here is that we're distorting these songs over day time scales. So the bird has the capacity, in principle, to learn that his target syllable has a high probability of error. So basically, if we're targeting syllable C, he learns that his syllable C kind of sucks. And if that's the case, then he could be comparing his song not just to a fixed template, but rather to his own past song, right? So to test this, we did experiments where we manipulated the probability of distortion across different syllables in the same song. So here, we're distorting syllable B half the time, and we're distorting syllable D only 20% of the time. So if the bird can track the quality of it, the predicted quality of his own song, then right before syllable B, right before this target time, he's got a high prediction he's going to make a mistake. And right before D, he's got a lower probability of making a mistake. Okay? And just to make this intuitive, I play tennis. My forehand's pretty good. My backhand kind of sucks. So if I hit a backhand in, it's more surprisingly good to me than if I hit a forehand in, right? And of course, I can't go out there and compare myself to Federer. I have to compare myself to my past self, right? So we're testing if the bird can do that with its song here. So on a rendition of the motif where we don't distort either syllable, the actual error is 0. But the predicted error is very high at the high probability target, lower, but non-zero, at the low probability target. So the prediction error is the difference between the predicted error and the actual error. So this would be the signal that you would expect in the dopamine neurons if they really are evaluating the song this way, according to a memory of past errors. And that's exactly what we saw. And so what we really showed is that the dopamine neurons are evaluating the song. They're evaluating each song syllable on the basis of how much it exceeded the expectations. Okay? All right. And I think this can be intuitive for us because this is obviously how we learn. We compare ourselves to our past selves. And that's really the learning signal you would want. Okay. So now the next question is, so we've got a dopaminergic signal in the bird that projects up to the vocal motor striatum, and it generates the exact type of signal that we know mediates reward maximizing stimulus response behaviors. So we got a circuit that looks like this in both the mammal, just learning to do something silly like moving a joystick down to number 2, and in the bird that's actually learning a song. So how could this same circuit, using the same learning rule, learn song? And so let me walk you through some speculations on this. So we learned, hopefully everyone in the audience can understand how you can learn to move down when you see the number 2 on the screen. Okay? You could also learn to move right when you see number 1, right? And you could learn a whole bunch of stimulus response associations. And if you're sitting there on a screen with a joystick in your hand, and this sequence of stimuli rapidly appeared on the screen, and you were well trained, you would just move the joystick in sequence, right? And rather than having a 4-dimensional output like a joystick, you can imagine you have piano keys as your output, maybe 14 piano keys. And so really what you need to do, the essence of learning a song is learning what combination of keys to press when. Now for this whole process to work, what you need in the brain is an internal representation of time that would sweep through time like a sequence of stimuli in the world. But it's not in the world, it's in your head, right? And it's been known for many years, since 2002, that there's a structure in the bird's motor cortex called HVC that does exactly this. So what's shown here is a song, and this is 250 HVC neurons recorded in the same bird in the lab of Michael Fee, my postdoctoral advisor. And shown here in yellow is when a given neuron was active in the song. And you'll see it forms this, looks like a line here. What that is a domino chain. Each neuron discharges a brief burst, about 10 milliseconds in duration, at a precise moment of the song, every time the bird sings that song, and it doesn't discharge at other times in the song. Okay? So what you've got is you've got representations of time tiled through the song. And that's exactly what we need. The other thing we need is piano keys. Now, for many of you, neuroscientists in the lab, you'll know that one of the core features of motor system organization is topography. Topography is just a word for mapping. And all mapping needs is that you've got these 7 muscles in the syrinx. You've got a motor neuron group in the brainstem that innervates these 7 muscles. And it's likely it is the case that the lateral part of the motor neuron group will innervate this key. And the medial part of the neuron group will innervate the low pitch key. And then in between, you've got the keys in between. So there's a mapping of the surface of the piano, if you will, in the brain. And this topography can be conserved throughout projections, and it is conserved in the topographic organization of the song system. So you can actually think of these basal ganglia and downstream thalamocortical circuits as being laid out in space like pianos. Okay? So you've got a representation of time that connects to piano keys, and you've got dopaminergic input coming in that can basically, through dopamine modulated cortical channel plasticity, link up discrete time steps to discrete actions. So here you've got a sensory stimulus number 2. You can learn to move down if that's the best outcome to get food or juice. The exact same circuit, I'm speculating, can link up a representation of time to the appropriate action in order not to get food or juice reward, but to get your own internally assessed performance quality signal out of the dopamine system. Okay? Does that make sense? All right. So the next question we want to address is, who on earth in the brain is telling dopamine neurons during singing to burst or pause? Right? So in classic tasks in mammals where the animals are getting food or juice, we know a lot about it. You've got all these limbic circuits that are associated with emotion and motivation and hedonic functions that project into the dopamine system to carry food, juice, reward signals. But what could be projecting into the part of the midbrain of the bird that's generating performance evaluation signals? Okay? So to address this question, Radon Chen, a graduate student in the lab, working with Pablo and Andrea and some amazing undergrads, Alex, Tori, Archana, and Kamal, set out to get at this question of, like, how do dopamine neurons know what sounds good or bad in the bird? Okay? And so this is, just to bring us all zeroed in on the same question here. We want to know who's telling the dopamine neuron to pause after the distortion? Who's telling the dopamine neuron to burst when there's no distortion at a time step of the song where the bird might think his predicted quality is low? Okay? So, previous work by other groups, and especially Dave Perkel at the University of Washington, had done anatomy, basically figuring out who in the brain is connected to who. And they identified 2 main inputs to the part of VTA that goes to area X. One was part of the auditory system, a structure called AIV, and one was a part of the ventral basal ganglia called the ventral palatal. And Michael Fee's lab had shown that AIV neurons that project to VTA burst following distortions. They send an excitatory projection into VTA, and what Radon and Andrea found is that if you record VTA neurons that go to X and stimulate AIV, you drive pauses. So they basically found it's very likely that AIV is poised to drive these pauses when there's a distortion. Now, what we were interested in is who's driving this burst of activity, especially because it's scaled by a memory of errors. And I think that's probably the most interesting thing going on here, that the bird is keeping track at each moment of his song of not just how he just did, but how well he thought he was going to do. Okay? So we focused our energies on the structure of the ventral palatum. No one had really looked at it before because it's outside the song system. It wasn't really thought to have anything to do with singing. So Tori, an undergrad, lesioned VP as part of her honors thesis project and found that birds couldn't learn to sing. So that green-lighted us to go invest a lot of effort to recording signals there. And we specifically wanted to know what signals come out of VP and go to VTA. And can they explain this signal when performance is better than predicted? Okay. So we recorded from VP neurons that project to VTA, and here's about half of the neurons that go to VTA do this. So this is the activity of a single neuron, a line of song. Here's the undistorted rendition. There's a distorted rendition. Here's the raster. And what you'll notice is there's a little pause in firing that you see right here, right before the time of the song that we had been distorted. So we interpret this as a predicted quality signal. Does that make sense? Okay. Other neurons that went to VTA did this. So again, this neuron is bursting right before the time of the song where there had been a lot of errors. This is a predicted error signal, right? This is like, imagine you're playing a song and there's one part of the song that you always screw up because it's a hard part of the song. As you're coming into that part of the song, you're like, oh, I'm about to screw up. There's that feeling. Maybe you guys can understand. You know you're about to make a mistake. You don't know. You have a sense that there's a high probability you're about to make a mistake. I think intuitively a lot of people can understand that, especially if they're musicians. And Pavel, who recorded a lot of these neurons, was a musician, and we had a fun conversation about that. So this is like you're about to mess up signal. And importantly, these are two prediction signals that you really predicted quality, predicted error that you would need to generate this. So we were really amazed to see these signals in the brain. They also have a very cognitive field of them, which is probably more than many people thought the birds were doing in learning their song. Now, other neurons we saw exhibited prediction error signals. Here's a neuron that burst right after undistorted renditions, but it didn't do that following distorted renditions. So this neuron actually must have auditory error information too. And here's yet another neuron that does the opposite, that pauses after so-called successes. So this is a complex data set. We're seeing a lot of mixed signals that we can imagine are all useful for computing prediction error. We don't quite know how, but we're seeing you could combine these signals to generate this. So I'm going to lay out some questions we address next, which is like, the VP had not really been studied before. We had no idea who's telling VP neurons how to do these signals, right? So in the same logic, you record VTA, you see these interesting performance evaluation signals. So where are these coming from? You go to VP, you see these interesting predicted error signals. So where are these coming from? And this is one of the ways I was trained to do systems neuroscience. You just track signals as they propagate through circuits, right? And so we wanted to know where these signals could be coming from. And so to do that, we injected retrograde tracer into VP to see who's projecting to VP. And to make a very long story short, and many hours of work, we found, among other things, that there's a filament structure called UVA that projects to VP. UVA had long been known to endow HVC with its timing information. This is a part of the motor thalamus that we know encodes time step like HVC. So great. VP needs to know what time it is in the song in order to have temporally specific predictions. So we needed that timing input. We also found that AIV, the same AIV neurons that project down to VTA to drive that error signal, collateralize into VP. So VP gets a copy of an auditory error. Okay? We need that too, because if you remember, this neuron is discharging differently on undistorted and distorted renditions, so he has to know about auditory errors. He also has to know what time it is in the song, because someone's driving that burst just at that time. And he also has to know what the probability of error was, because it's bursting right after a predicted error did not occur. Okay? So we've got, and the third main projection was the same VTA neurons in Vikram recorder that go to area X that encode prediction error, they collateralize into VP. So we came up with a speculative model that said we can explain all of the signals we see with just these three inputs. And I'm going to walk you through that, and it's going to be about 5 minutes of a pretty dense, deep dive. So if you tune out for a second, that's all right. I'll come back and I'll do it afterwards. Okay? So you have to hold a lot in your brain here. Okay. So I'm going to just summarize all the anatomy of the signals and try to put it into like one coherent picture. Okay, so let's just start. We've got Vikram's dopaminergic error signal. A burst when the song was better than predicted, and a pause when the song was worse than predicted. This signal goes into the vocal motor striatum, which is organized like a piano, and which gets timing inputs from HVC. And we learned that if dopamine neurons burst right after hitting this key at time step 2, then you'll strengthen the connection between time step 2 and that key. And that'll enable you to do the best action at time step 2. Okay? And then we wanted to know where this signal comes from. And we knew from past work from Michael Fee's lab that neurons in the auditory cortex that project onto VTA burst after auditory errors. And Radon and Andrea found that this signal gets inverted by local inhibitory circuits in VTA. So this burst here can get inverted to drive this pause here. Okay? So we think that's being driven by AIV. But next we wanted to know, okay, who's driving this burst? In order to do that, you need to know about predicted quality, not just if you made a mistake. You need to scale that mistake by your predictions. So we looked at the structure of VP, Tori lesioned it, birds could learn to sing, so we started recording there. Well, first we did some anatomy there. So who's going to VP? We found that UVA goes to VP, and UVA encodes timing, just like HVC does. In fact, UVA projects to HVC and gives it its timing signals. So, then we finally also found that the same VTA neuron, this signal that goes here, also goes here. So by the exact same process by which you can weight the connection strength between a time step of the song and a key, according to past error, here you can weight time step alone according to past error. So imagine you've got a 3 time step song. Time step 1 and 3, you're good. You don't make mistakes. Time step 2, you have a high probability of error. But what that means is that the discharge of neuron 2 will be coincident with these pauses that are being driven by the auditory system. If you have a dopamine modulated plasticity, which is universally observed in the brain, you could weight these synapses according to their coincidence with phasic modulation of the dopamine signal. And then you can get basically error-weighted timing. So it's a representation of time that's weighted by your past error. Does that make sense? That is equivalent to a predicted error signal. So we think this process can explain how you can get a signal like that. Right? So time step 1 and 3, you're good. Time step 2, you've got a big history of errors. You've weighted that synapse, so time step 2 comes in and boom, drives that first right before you're about to make a mistake. Now, if this was the only signal we saw in VP, we'd be happy as clams, because this is predicted error. In order to compute prediction error, all you need to do is subtract predicted error minus actual error equals prediction error. We could go home. And I wish it was like that. But biology is never like that, right? So something deeper is happening that we don't yet understand, because what we see is, we see predicted error, we see predicted quality, we see auditory error that we think comes from AIP, right? We also see prediction errors. So the same process that we think results in the computing this signal here can also happen up here. So the big conclusion is we don't yet know exactly how these signals are combined to create this prediction error signal that comes out of the dopamine system, but we know that we have all the ingredients for it. All of these signals can be pretty simply and linearly combined to produce this. And as an aside, there's only been one other study ever that looked at the signals that project 2 dopamine neurons, and that was done in mice by Naoshida's lab, and they imaged neurons that were projecting the VTA dopamine neurons. And they also found, in many inputs to the dopamine system, mixed signals like this. Even though in that behavior, all of the dopamine outputs did the same thing, which was prediction. So somehow, this circuit is like homogenizing its inputs to create this signal, which is really the perfect signal for learning. Okay. All right. It was pointed out to us by Josh Dubin, who's on Radon's committee, that this architecture we came up with is literally identical to classic actor-critic architectures from long ago when people were looking at how dopamine reward prediction error signals are constructed in foraging contexts. Because basically, you don't need to understand the details of this, but you've got a dopamine signal that trains an actor to make reward maximizing stimulus response associations. And that same dopamine signal goes to a critic whose output is not the motor system, but whose output is back to the very dopamine neurons that trained up in the first place. And here you're weighting stimuli by their past errors or rewards, and then you get a stimulus-dependent reward prediction. And here you're actually converting that, because you've got action channels, into reward maximizing stimulus response association. Okay. Good. Now there's a problem. And this occurred to me while walking to work one day. Animals can exhibit place preference on the basis of having dopamine stimulated. So let's say you've got a rat in an eye-shaped maze, and he's just wandering around, and every time he goes up here, you stimulate dopamine neurons. After a little bit of time, the rats will hang out here. Okay? The reason why that's a problem is because birds vocalize as they do other things. And you wouldn't want a bird who hits the right note next to a snake nest to develop preference for that place. Okay? And this is a universal quality of vocalization. I think maybe it's something that's unique about vocalization is that animals vocalize as they're doing other things. Right? I mean, even this example, he's also learning how to pick up pages and turn them as he's babbling. right? So if he happens to babble a right sound, it shouldn't reinforce the way he grabbed the page. You want to keep these systems segregated. So in the interest of time, I'm going to make a long story short. We hypothesize that actually the way the brains are organized is into different agencies. So within an individual animal, different systems in the brain have different objectives. The navigation system wants to go places that are safe, filled with food, reward, and mates. That's the objective of the navigation system. The vocal system has a different objective. It can't control navigation. It controls vocal output. The objective of the vocal system is to copy a tutor song, right? And so people often think of global reinforcements, but you can imagine that reinforcement signals are isolated, that a singing-related vocal error in the auditory domain will only reach the vocal motor system, and something like a strobe light, or something that freaks out an animal, will result in a preference to avoid that space. But the bird won't be able to change the way it sings to avoid that strobe light, right? So long story short, we found that birds have different distinct agencies in the brain. Birds, we set up a cage where we could detect how the bird was singing in real time and what perch they were on. And we asked if they could change the way they land on change which perches they like on the basis of distortion sounds or strobe lights. And then we asked if they could change the way they sing on the basis of distortion sounds or strobe lights. We found is that a bird couldn't change the way it sings to avoid a strobe light. But a bird could definitely learn to not go on perches where they get hit with strobe lights. And birds actually like to hang on perches where they got these little distortion sounds, even when they were singing, even though their vocal motor system did everything, like change the pitch to avoid those distortion sounds. So basically, there's no such thing as a bird likes this. The bird has multiple agencies inside of it, and each of those agencies can have its own objectives and its own reinforcement signals. Okay. Let's see, I got about 10 minutes. Okay, that's cool. All right, so, of course, it's often the case, especially if the bird's in a good spot, that it's not being attacked by a snake, but it's singing to a mate. And what he really wants to be saying is, I'm really fit. He wants to convince her that he's fit. And then magic happens. And all of the experiments I talked about where the birds were learning to sing and evaluating their songs were when the bird was alone. Okay? But these birds sing to females to convince them that they're fit. And I just want to show you a video of this that I got from Mike Long, but we see this a lot too. So here's a bird hanging out, and then you're going to see what happens when he sees a female. It's a very powerful behavior. That male bird sees a female and he orients to her, follows her around, and he sings faster and in a much more stereotyped way than when he sings alone. And most people in the field before we got into dopamine, not most, but many people just thought that the reason why this dopamine signal was going to area X was all about the female. It was all about, oh, this is going to be what makes the bird sing to the female and sing more stereotyped ragged songs. And there was some reason for that. First, when a bird sings alone, its song is variable. It's like he's kind of practicing, he's playing around. When he sings directed song, it's all business. He's in performance mode. And he's just singing ABCD, 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 with very little variability. And it was shown that if you block dopamine receptors in area X, you don't get that normal reduction of variability that's driven by the female. So it was thought that what dopamine was doing in area X was mediating this transition to performance mode and the corresponding reduction of vocal variability. But it had never really directly been tested, and with Vikram's really unique ability to record these neurons, we felt like we could get at this question. So what he did was, he recorded from dopamine neurons in a pretty clever experimental design. So first he would have the bird sing alone. He'd be distorting the target syllable as you saw before. So we get the air signal, a little burst after distortion, a little pause after distortion, burst. And then what he would do is, while holding that exact same neuron, he would show the female. And there were many possible outcomes of this experiment. The one that most people in the birdsong field, and maybe more generally, would think is that, oh, when the female appears, the dopamine neurons are going to get activated. Right? I mean, there's a lot of popular press about dopamine neurons activated by mating context, but there's no evidence that dopamine neurons are activated in mating contexts if you consider spiking activity of neurons as what constitutes evidence. So what he found was, when the birds are singing alone, you get these nice error signals that we've already found. So we replicated that with an independent data set. And then when they sang to a female, the firing rates did not change at all. Okay? But what happened was the error signals in 5 of the 7 neurons totally went away. And then they were kind of still there in 2 of them, but a little bit. So it's like the dopaminergic error signal was gated off when the animal transitioned to a performance mode. As if it's no longer even attending to its own song anymore, and maybe to something else. Does that make sense? And to our knowledge, this is the first example of a dopaminergic reward or error signal being gated by like a social context like this. And so this is currently in prep. And I want to, with the last 5 minutes, talk a little bit about what we're doing in mice, and then parrots too. Okay. So, first, okay, so I'll get to future direction. So future directions. In the bird, what we're doing is we're realizing by following up on inputs to VP that there are lots of other structures in the brain that are not supposed to have anything to do with motor sequence learning or birdsong that we're starting to think are very involved with the imposition of valence on performance errors or prediction errors, such as the lateral habenula, which is Andrea's graduate project, and STN, which is Anandita's postdoctoral project. So we're starting to get pulled into these parts of the brain that I never thought I'd be working on. That's funded by agencies like National Institute of Drug Abuse and National Institute of Mental Health that haven't really had much evidence of being involved with motor learning, but I think all of us can understand if you double fault on match point, it's a very emotional experience. So it shouldn't be surprising that a lot of these circuits or so-called parts of the limbic system are going to be used to make the most of those error signals for the purpose of learning. And maybe that's what emotion is really all about, right? That's my opinion. Okay. Next, we're getting interested in parrots, partly because Jack Bradbury's been nagging me about it. But there's such cool stuff going on with parrots. I don't have time to talk about it. But, so as many of you know, parrots, such as parakeets, have an amazing vocal repertoire. They basically hang out all day and have conversations. And they're mimics, and they can, zebrafinch sings like 6 syllables. These guys are off the charts. And what's so cool about their neural circuit is that while the subcortical pathways in the brain that we've been studying are basically the zebrafinchy or songbirdie, parrots independently evolved their vocal learning capacity. And they've got this really different cortical circuit. And basically, here's the rub, is that whereas in songbirds, many of these circuits are feed-forward, HAC projects to RA, which projects to motor neurons, projects to motor output, and you've got like this domino chain. In humans and in parrots, that's not the way the vocal system's organized. Everything is in a loop. Everything that you can imagine is in a recurrent loop. And with recurrent networks, you get a lot of more interesting time-dependent dynamics, much more flexibility, and I think this is what's going to be behind their outsized vocal capacities. So we're starting to get into what is different in the signals that propagate through the vocal motor regions of parrots. Okay, and we've got a new undergrad, Eugene, working on that with Radon. Okay, and then this is the part of the talk where I talk about Tenure. When I was leaving MIT to come here, and I was asking Michael Fee for advice, which I rarely did, I was like, oh man, starting my lab, I'm pretty nervous about this. And he said, all you need is that first graduate student. He said that. And I was incredibly lucky to have Tasia my first graduate student. Tasia built most of the songbird rigs before he started working on mice. And then what he's done is he's sort of built a whole lab in my lab. And one of the first big grants I got, the Innovator Award, when I got that grant, it was this weird feeling. It was like the first time I realized that I got credit for something I didn't know. I was like, this was Tasia's grant. It was all his data, a lot of his ideas, and then I got the credit for it. And I want to just pay that back a little bit. So what's Tasia been up to? He's been created, as I said, a lab in the lab, and he's worked really closely with Sam Whitehead from physics, and some new students he's working with, Brendan, Alice, our technician Brian, and many undergrads. And he basically built up ways of measuring mouse behavior that we think are really important. Mice are a really important model system for neuroscience. There's a lot of genetic tools. You can turn on and off different pathways in the brain really easily. And one of the things that we got into was reaching, in part not motivated here. Reaching is an interesting behavior. It's typically studied in monkeys. And what Tasia did is he built this system, whoops, you're like, what the heck is that? He built this system where he built this joystick with a touch sensor, and we can basically monitor mouse four-limb trajectories with micron millisecond precision as we turn on and off different parts of the brain and see how its reach patterns are affected. See that here? And this is laser going in the brain to turn off different pathways, and this is the whole thing's not playing right. But basically, it's because these files are so big. We can see how the kinematics of the limb are affected in a center out reach task as we play with different parts of the motor system, which is extremely complex. There's cerebral circuits, basal ganglia, brainstem, cortex. There's a lot of work to do. And he's made some important discoveries about the role of cortex in forelimb movement. And then what we realized on the side, what Tenure realized while watching his mice do lick tasks. And it's one of those things where it's obvious what you hear, but you just don't think it. Many people look at mouse licking these days. The reason why is because if you can head fix a mouse and get them to read out a choice task, then you can bring incredible tools for neuroscience. You can record from thousands of neurons in the brain of a head fix mouse. You can image all the neurons in like millimeters of tissue. Not all, but many of them. There's incredible tools. But the mice are doing these kind of, what does a mouse do? Right? It licks left, it licks right. And so there's been a lot of work looking at mouse licking. But no one had actually looked at mouse licking. They were looking at the ability of a tongue to contact the spout. Which is kind of like, if I'm studying reaching, I'm going to look at the ability of my hand to reach a mug, and I either do or don't. But all the interesting kinematics of the tongue were invisible. And Tasia and Sam, in a fit of what seemed to be like manic stupor and productivity, stole Itai Cohen's very expensive camera, brought it over to our lab, and set up this tongue imaging system. And they imaged the tongue at like a kilohertz. And then Tasia built this deep convolutional neural network to track the tongue. And when you look at these videos, you realize the tongue is just like a limb. It reaches for things, it grabs them, it makes mistakes, it corrects for them, but it all happens in like milliseconds, right? And it's a really interesting behavior that Brendan in your grad student is following up on, and I'm getting really interested in. And then, so here's just an example of the data you can get. There's a paper that's we're putting together right now. You can track the tongue with millisecond precision as it moves looking for targets. And it just does exactly what your arm would do. Let's say you have a spout over here. It's trying to find it. It reaches out for the spout. It's like, where's the spout? Oh, there's the spout. And it just grabs the water. Right? So by studying tongue and limb, we can hopefully distinguish between effector-specific modes of operation and mechanisms from general principles. Okay. So here's my summary. Okay, so the big idea I want you to walk away with is that, based on our findings in the bird, the same brain mechanisms that evaluate reward outcomes can be redeployed or repurposed to do internally guided motor sequence learning. And not just the same dopamine signals, but actually the same circuit architecture that constructs those dopamine signals so that they encode prediction errors. In other words, these neurons aren't just activated by rewards. They're only activated by better than expected outcomes. And they're not just suppressed when something bad happens. They're only suppressed when something worse than expected happens, right? There's a big difference there. And it highlights the fact that what the brain is a prediction machine, and we're going to keep those predictions accurate and learn from prediction errors, okay? I talked about how a bird has many agents inside of it. And I talked about how these dopaminergic error signals can be gated on and off by different performance versus practice contexts. And then finally, I was trying to think as I reflected on what this talk is, I was like, what am I? And I think what we do in the spirit of this department is comparative systems neuroscience. So, you know, we want to study sort of similar questions in the context of different animal models and also different behaviors. And I think that's important because it's really the only way, you don't have to all do it in the lab, but you should at least be aware of, like, if you're studying principles of reaching, you should know if they also apply to licking. And if you're studying principles of vocal learning, you should know if they apply to, like, reinforcement learning in mice. So I think having this kind of approach is useful for distinguishing sort of idiosyncratic mechanistic solutions to a problem to actual general principles. And with that, I want to thank you all for coming. My heart is really filled by this room, I have to say, seriously. And especially thanks to this crowd and these guys right here. Maybe 3 more. How about that? In the next phase.
We have enough mugs.
I can use some more mugs, Rob. And thank you to all the support I've had at Cornell, both from the grad students, postdocs, and undergrads in my lab. You know, Tom, Joe, Rob, and Craig have all helped me with the grant process. I remember the first grant I gave to Tom Seeley. I thought it was so good, it came back and it's like all red. And I was like, wow, Tom's a good writer. And I learned a lot from that process. Joe basically wrote my R1 Ames page, so if anyone needs to get an R1, get in touch with Joe. They might not be here, but the people who care for the animals, they're amazing here. So Rich Moore, Tim Van Dusen, Steph Pearson, obviously Sonia, Terry, Lori, Kathy, Sarah, Stacy, John Howe, Adrian, and Julie, they make the whole department work. And thank you all for coming.
Must have questions. This is our time.
Hey, Dave. Hey, thanks.
Okay, so Jesse, I have a question. You said that birds can't really relearn to sing, that if they go on a perch that they don't like, they won't change their song. They'll choose perches, but they're fixed in the song. Is that right?
So what we did was, I had to race through that, so I'm glad you're asking, because it wasn't clear. What we did was, let's say the bird sings A, B, C, D. And every time he sings C, he can sing it a little bit differently. Sometimes he sings C, and sometimes he sings C. A, B, C, D, A, B, C, D. And the way that those classic distortion experiments were done is that you deliver the brief 50 millisecond snippet of sound, contingent on the pitch of a target syllable, then the bird will learn to change the way he sings that syllable to avoid that distortion. So you can drive the pitch up or down with a brief 50 millisecond noise burst. So we knew that a brief noise burst gets into the vocal motor circuit to drive behavioral change. We did that exact same experiment. Instead of playing a 50 millisecond snippet of sound, we did a 50 millisecond snippet of flickering lights of high intensity. And the birds never learned to change the way they sang their syllable to avoid the strobe with their song. But they rapidly learned to avoid perches that elicited the strobe light. So our logic was that whatever reinforcement signals are being driven by the strobe light that can reach the navigation circuits in the brain cannot reach the vocal motor parts of the brain. And in some ways, there's classic psychology work. Like this is, as a vet, you probably know this, like Garcia, the work by Garcia, like, I forget exactly how it goes, but like, they can learn if they eat something that makes them nauseous, they can learn to avoid that food. But that nausea doesn't make them learn to avoid some spout that they got the food from. So it has to do with the way evolution has wired up these circuits that specific agencies in the brain, by definition, have specific objectives. And to meet those objectives, they get very specific reinforcement signals. Right? That's kind of the idea.
So the reason I ask is because if you wanted to get the tutor song right always, if you learned a tutor song in childhood, and you wanted to be good at that tutor song, it seems to me that it would be easiest to always compare your performance against your memory of the song versus memory of your own failings. The fidelity might be best if you were always comparing against the template versus iterations on how you did it in the past.
So yeah, you're right. No, you're right. So I must have, the tutor song memory is still there.
Right.
The nature of reinforcement it's called an advantage function for those machine learning people. It basically, the question is, was I closer or farther from my target than I thought I would be based on my past history? That's the question, right? So the target's still there, it doesn't just disappear. That's the anchor, but then the learning signal is basically, is what I just did closer or farther? And then if it's closer, reinforce it, right? And if it's farther, then extinguish that, right? So you're still, yes, the tutor memory's still got to be there. And actually, one of the reasons why I glossed over it is we have no idea how that comes into play. It's probably up in the auditory system, upstream of that error signal that's coming out of that structure AID. Hi.
Hi. So, this is based a little bit off that. So initially I was going to ask about external stimuli. A lot of these things are strobe lights are given under the same food, right? But what if you change the nutrient composition of the food? And does that alter the response to the external stimuli?
You mean in these original experiments?
Yeah.
So, I mean, that I don't know.
Thinking about B vitamins or things, glucose, caffeine, these things alter our performance. And I'm curious to know whether nutrients or internal stimuli affect responses to external stimuli in performance or practice.
Yeah, we haven't looked at all. We haven't looked at that. I think that the classic psychology literature that you might know better than I do, looking at nutrition and performance. I mean, and that's got like in kinesiology. There is an idea that like one of the reasons why the bird song is supposed to be an honest signal of their fitness is I mean, I don't know if I believe it all, but like it's supposed to indicate the health of the animal. And there's this idea that if you give a bird bad nutrition in its childhood, its song learning, it's reflected in how good its adult song is. I mean, that's actually the canonical way that people think about why bird song is a signal for mating. There's supposed to be some honesty to it. Yeah. I guess Mike, you were first, and then I guess.
So I'm just curious, if it's just wild speculation, for a bird like a mockingbird that doesn't have a crystallized song that it's targeting, could you speculate maybe one or two ways that you think the circuit would be different?
Yeah. Well, I think the circuit could be the same. It's just a slight tweak.
How it might change.
Yeah, I thought a lot about. Okay, so we thought about that. We talked about that in the lab. So basically, what you're at, zero finches are special because they just go A, B, C, D, A, B, C, D, A, B, C, D. But the essence of what they're doing is, and maybe this is best, easiest to articulate with the image of Evan babbling in your mind. So you're producing behavior, right, with your motor system. And aligned to that behavioral production, you've got sensory targets, right? And there's an intersection between the two, right? So when he's saying really fast car, his tongue and lips are moving in such ways to make these sounds, but in parallel, his auditory system is simulating what he wants to hear, right? So this is actually what Michael Fee talked about a couple weeks ago when he came to give his neurotech talk. He was talking about, what I didn't talk about is HVC gets inputs from the auditory system and projects back to the auditory system. So that clock, that domino chain, doesn't just go to the motor system. It actually goes back into the auditory system, right? So the auditory system knows at each time step, it can learn at each time step what sounds it's about to hear. The auditory system also projects into HVC, so they're in a loop, right? So you can have a sequence of auditory targets, a sequence of motor actions, and you can, in principle, compare the two. And it's probably happening in cortical loops that connect HVC with the auditory system. And that's actually kind of the focus of what Michael Fee's research is now. Michael Fee's on postdoc. Now, the question is whether or not, and then when you look at it that way, it doesn't really matter how limited the song is. It doesn't matter if it's one syllable or 100 syllables. It just has to do with the alignment of the auditory target with the motor system.
Well, my point is that in some of these sport species, there's not like a set alignment of songs you can do it all the time. Right? If you're in the same sequence, right?
Yeah, okay, so when I say la, I think to myself in advance of saying la, I have an auditory simulation of la. When I say baka, I just said that, but my auditory system, even as I was saying it, had a prediction and an intention of saying baka, right? So I guess so what's the...
I don't know anything about it. I was just curious. It's
I
not
guess
like a secret question to take down.
It's not what?
No, it's just like I was just curious, right? Like they're coming up with some cases they're coming up with novel sounds, in other cases they're
Okay. Novel sounds a different question.
they're copying other things.
novel sounds is a different question. Novel sounds are, at least in zero pitches, known to be driven by a different structure called L-man. So HVC has got this sequence, it's banging on the piano. A different structure called L-man also projects the piano, and it has a lot of random dynamics. In fact, it's there to have random dynamics. So in a babbling bird, if you inactivate L-man, you take a babbling bird, and all of a sudden it goes chwah, 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 it loses its variability. So this was a lot of my postdoctoral work was about, was how does variability generate? So I imagine that mockingbirds and birds like starlings with multiple variable songs are having a larger L-man contribution to the piano than HVC, right? So, but then that's not a full answer to the question because it gets back to the first part, is they still have to be able to control L-man in some way so it's not totally random. And that's not to happen through, so the pathway number was HVC to X, and then to thalamus up to L-man, then to the piano. So the L-man activity that's variable can be biased by the process I talked about. It doesn't answer your question in full. We don't fully know how mockingbirds sing. This is some ideas.
Let's take one more question.
Yeah, one more question. Okay. I think Andy, you had a question.
So my question is about the signal coming out of UVA.
Yeah.
So you said that the timing information comes from UVA to HPC.
Yeah. Yeah.
What is the nature of that timing signal? How much timing information is really needed?
Good.
To activate?
Wait, how much time information is needed for what?
Really to turn on the PTA stuff, etc. I mean, how specific is that timing information?
Okay.
The details of
So
HPC.
the recordings from UVA were multi-unit, so we don't know for sure. We know the timing information is there. What I think is happening is that HVC has chains, and each neuron owns about 5 to 10 milliseconds of that chain. And it's been shown that chains are as long as about a syllable, or for complex syllables, it can be multiple chains. UVA kicks off the chain goes for like a syllable. Then HVC through RA, then down into the brainstem, and then back up to UVA, to the contralateral HVC, kicks off the next chain. So if I'm the song, let's say I'm singing with my right or left hemisphere. A, B, C, D. So actually that chain, although it's continuously covered in one HVC, the production actually is, there's this rapid interhemispheric switching at chain intervals. And so the idea is that what UVA's doing is kicking off the chain. So the prediction would be that inter-burst intervals in UVA are inter-chain onset intervals. And that what the unit of evaluation is that dopamine's caring about is it's evaluating a chain, which is ballpark 100 millisecond duration, which is ballpark the inner spike interval of a dopamine neuron. So it seems like it's all temporally matched, right? You can imagine that UVA kicks off the chain happens, then the next dopamine spike is evaluating that chain. Then meanwhile, there's another chain. And there's evidence behaviorally that chains are selected as units, as chunks, right? So this is asking, really what you're asking is like, how is behavior chunked? Behavior's not chunked into 5 millisecond intervals. It's chunked into 100 millisecond intervals.
Yeah, I mean, essentially UVA's coding that particular piece of it.
Yes, just a chunk, either a syllable or a complex syllable, like a couple of units there. Yeah. Okay.
Grad students, there is lunch.
How are you? You're coming to the meeting, right? You're going to be giving a talk? Yeah. Yeah, I think Jesse's going to come too.
Oh, it's just so at the final for last semester. Okay. And I responded, and I was like super interested, and then responded. For other things, somewhere.
Yeah, right.
We should. We don't get the vote, by the way. The Maritime don't get the vote. No, the Maritime do not get the vote. What's clear that Bob wanted to get the invitation to the meeting? Was there a meeting scheduled? It hasn't been called yet, but Bob just told Sandy that we don't get the vote. But maybe, you know, maybe that's just done. I get the vote, but it's yours. Amazing. He's really done well here.
So much beyond.
No, I think that's right. I think we're hitting it with the
Yeah, you're right. Imagine Marlon. I wish you were here to hear it.
He would totally.
Even Fernando might.
the spikes that are happening outside of we've
Just about anything that you can imagine. Layer upon layer of it's all there.
And as he strength, he looks at them. That is so much different from some. Which I got to say something. Do you want to chat and then I'll be right back?
Oh, yeah. Couple years ago, yeah. His picture, generalizing across systems, like Curtis Bill, the kind of Curtis Bill used to do. Asking huge questions, and then going into the circuit.
But it's not just a regular if that's you, it's not there's some sort of there's extra reward on just like on the right. It's like
well, so the baseline rate does not look carried over. They pull the ratio over baseline. So the dopamine receptors, one that's sensitive to bursts, and T1s are sensitive to bursts, and they have very low affinity, but they're fast at timing.
Right.
So they're sitting there, and they don't find dopamine, and then they burst with dopamine, and they find other receptors, but that has other dopamine receptors too, they have very high affinity dopamine, and they're usually bound. And then when there's a pause, they unbind, and that's the cell. So they've got different mechanisms of pulling this out from this. And it's thought that just baseline rates don't really produce much downstream. Like the tonic rate, the fact that they just plug away at like 10 hertz. That doesn't mean anything. It's when they dip down and peak up. The transit.
if there was no signal though, they wouldn't sing, right? They wouldn't hear us sing. There'd be no motivation to sing. Baseline. Is that accurate or not? Like, you don't always sing in the shower. You sing because you like to
Yeah,
sing.
I'm not sure. Well, not these ones.
Okay,
So
all right.
actually, I can make it clear. It's only a small subset. Just the ones that
Why don't you grab your...
Right. And it's only about learning. It's not about singing.
I want to know how you make this is more regulated by the
Pretty important. Hi, David.
So you have
Probably like somebody who's, I mean, he's been in a lot of races, right?
That's why I said to Greg, I said, it's like Jesse's out there. One of them, you know?
Where did you go?
Ice cream. Ice cream.
It sounds like Chris is going to take an ice cream.
You want ice cream, Sam?
Oh, actually, we're not going to
I got a picture.
I will, I
That's that mug swap. I'm sure you'll find out where it is. It's in some community college.
Yeah.
There's one other study that's currently in review by this.
So yeah, real people. That was great.
Thank you.
So you have 3 kids? Yeah.
When you get with the high grades work,
Well,
imaging don't get.
some things we don't do very well.
Like
the
yes, we got no, it's really
There's
nice.
a lot.
Ease with it all. Thank you. Very, very You asked some big questions.
Very respected. I'm sure you're super proud of him. And he's just a fabulous colleague.
Probably not.
It's been so fun to interact with him. I'm a big fan birds and because we do bird songs. So.
Maybe if they're really fast, that's just fast flyers. Like,
He is at home too. We still keep him around.
And his teacher that he was leaving school for.
Okay, so I guess.
What did you tell your teacher? What did you tell your teacher? Sandy.
I'm sorry, I don't know your name.
Sandy, what did you tell your
Sandy.
teacher?
We've met before. I'm sure we've met before. Because that's Sandy. Yes.
I agree.
What did you tell
I'm sure we got that. I can't remember exactly when. We were at the lab of ornithology maybe when you first came.
You can get permission to get out of work soon
Me?
enough.
No.
Oh, I can't forget my mom.
Great job, Jesse.
Thank you so much.
Would have liked to have heard more about the parrots, but of course you didn't have time.
They'll get
What's
there.
that?
They've only just arrived in the lab.
Yeah.
What's that?
We're looking forward to hearing more about the parrots.
Oh, yeah. I mean,
Yeah.
Raymond was just saying that we're going to record from them as they're talking to each other.
Do you have Evan's shoes?
They're upstairs.
Yeah, they're here.
Yeah.
He's not going far. So were you home last weekend when you had all these guests?
No, I was the one who was away.
You were away, okay.
He made room for me.
Oh, that's good.
I really enjoyed your family.
I know, it's fun, isn't it?
It was so cute to see your girls' Instagram. Of course,
Yeah.
I see Instagram. And to see your girls with my little boys, it was just
Yeah.
so
No, did the guys have a good camp?
Yes, fish, marshmallows.
There's a mug with coffee under your chair.
Oh, you know what? We should grab that. At least put it on the table so that it doesn't get kicked over. Can you grab it? I should grab this. This was Evan's water, too. Somebody will come back for it, probably. We can go around. There's a plate down there. We can help him clean up a little bit. Evan, you want any more of this water? Last chance for water.
No.
Going.
They left their pretzels.
Those were here when I came in.
Here,
go. I gotta go.
Wait, get Mama in there.